Hi, science friends. I want to wrap up this section on cell communication today, and we're going to talk about the cellular responses. So, we've really been diving into these signaling pathways, and next we're going to jump on into mitosis, but we're going to talk about these cellular responses today. So, before we do, let's have a little concept check here to make sure we remember what we've been learning. So, explain the terms phosphorylation and dephosphorylation. Those should be pretty easy. Adding a phosphate is phosphorylation, and then dephosphorylation would be removing a phosphate. So, I hope you all got those. What enzymes are in charge of phosphorylating and dephosphorylating? So phosphorylating is going to be protein kinase or a kinase enzyme, okay, which is a protein. So just kinase would be fine. And then dephosphorylating is going to be phosphatase, protein phosphatase. Okay, so hopefully you got those as well. Remember, both of those end in ase, so you know they are an enzyme. And then last, to what are the phosphates usually added or removed, and why is this important in signal transduction? So generally the phosphates are going to be added or removed to a protein. And that's important because when you add it, it causes a change in shape, which changes the function. Okay? And that's really important, and it can go on in a signaling pathway in that phosphorylation cascade that we talked about yesterday. Okay, and that's going to generally happen in the transduction step, mostly is where you see all of this phosphorylation happening. So today we are going to talk about the signal responses. So in a signal response, a lot of times what happened is protein activity is regulated. Cell division can also be regulated, and this next part of the second part of the unit that we're going to discuss is cell division, also known as mitosis. You may have been familiar with that that term. But signaling molecules that initiate cell division pathways are referred to as growth factors. So let's take a look at this diagram here. So we have our signaling molecule, also called a ligand, and this one's showing a growth factor. So it binds to the receptor, the receptor protein in the cell membrane. That's that first step, that reception step. And it causes that phosphorylation cascade where we activate all of these inactive protein kinases. We phosphorylate them. These are kind of called our relay proteins that we've got here. Okay, you'd have your second messenger in here to help with those steps. This is shown very vaguely. But that is the transduction phase. And then sometimes the transduction phase will go on to activate a different kind of molecule called a transcription factor. So we see this inactive transcription factor gets activated. You notice that a phosphate has been added onto it here, and you should also notice the change in shape. So it went from this bean shape to this kind of C shape. And that C shape there, our active transcription factor, binds onto the DNA, and that is the third step. That is the response. And so by binding to the DNA, it is either accessing the DNA and allowing for transcription of the DNA, or it can block access to the DNA and stopping transcription of the DNA. So if you remember, we talked about this very, very briefly in biochemistry, and we'll do a whole unit on this after Christmas. But the central dogma, it's called the central dogma in biology, is that DNA is used to make RNA, which makes proteins. Okay? This first step is called transcription. And the second step is called translation. Transcription happens in the nucleus, because that's where the DNA lives and it doesn't leave. Translation happens where the proteins are made. Where are the proteins made? You should say the ribosome. That is responsible for making proteins. You remember that from the last unit. So this is the central dogma. DNA is used to make RNA, which is used to make proteins. And you see that here. DNA is used to make a small segment of RNA called, which codes for a gene. And that gene codes for a protein. This mRNA would go bind to a ribosome and make a protein. Okay? And so this transcription factor binds to the DNA and it allows it to either causes it to synthesize the DNA or blocks the synthesis of the DNA. And synthesize, I mean either copy it to make like a whole new organism, to make whole new cells, or copying just a segment of it to be able to make a protein. Okay, and again, we will get all into this part of transcription and translation and all of that later. But for today, what I want you to know is that sometimes the signal response can be to activate a transcription factor, which causes genes to be transcribed and proteins to be made. Okay, because these transcription factors bind to the DNA and the DNA codes for proteins, essentially. Okay, so hopefully you can just manage that part for today. Let's move on. So this is another diagram, and I just want to use this to kind of check understanding. So we see our 3 steps here in our signaling pathway. You should be excellent at these 3 steps now. There's no reason that you shouldn't know them because I've numbered 1 to 3 about 20 times now. Okay, reception, transduction, response. So our reception binding epinephrine to our G protein coupled receptor. Okay, so that is one of our 3 major receptors that we talked about. Remember, we talked about the G protein coupled receptor. We talked about the receptor tyrosine kinase. And we talked about our ligand-gated ion channel. Okay, that when the signaling molecule bound, it opened the gate. Okay, and so those are the transmembrane receptors here. This one specifically that we're discussing is the G protein coupled receptor. What is our signaling molecule here? You should say that the signaling molecule is epinephrine. That's what binds to the G protein coupled receptor. Okay? When it binds, it activates this G protein. So we've got our inactive G protein that gets activated. Okay? Now, what second messengers do you see in this pathway? Remember, we talked about 2 second messengers yesterday. Which one do you see here? You should say cyclic AMP, or we referred to it as CAMP. Okay? That was one of the major second messengers that we discussed yesterday. The other one was the calcium ion, was an important second messenger. But here we see CAMP. Remember, we talked about an enzyme that converted ATP to CAMP, and that was one of those 4 enzymes that you wrote down on your note card yesterday. And you see that here as well. Adenyl cyclase converts ATP to CAMP. So there's that important enzyme that we talked about yesterday. Okay? Now, one thing that's really important that I want to talk about is this pathway is drawn very simplistically. Okay? When one epinephrine binds, one epinephrine can actually activate over 100 G proteins. Okay? You don't have to have one epinephrine for every G protein. And so this signal, if this is one branch off of the epinephrine, then it's got 100 branches. It's activating many more molecules. Okay? This is just one leg of it, or one branch, one arm of the signal pathway. That one epinephrine is going to activate many more G proteins, which all are going to follow this same pathway. Okay? So ultimately, the second messengers here, like CAMP, they generally activate a kinase. Okay? And those kinases go on to elicit a response. And the cellular response, what is that in this step? What is the final response in this pathway? Glycogen is converted to glucose. Glycogen is a stored starch that's stored in your liver. And it's broken down into glucose, which is the energy that your body can use to make ATP in the process of cellular respiration, which we'll get into really big in our next unit of metabolism. Okay? But the response here is glycogen is broken down into glucose. Okay? I also want you to see in this diagram that here we have one molecule. There's one signaling molecule of epinephrine here. Look how many molecules are here. 10 to the 2. 10 to the 4. Sorry, my diagram's gotten sloppy. 10 to the 5. 10 to the 6. Okay? 10 to the, I kind of marked through that one. Maybe it says 10 to the 8th. But the longer this pathway goes, and I talked about this briefly on the last day that we were in class together, the bigger the response gets. Make sure you write that down. The longer the pathway, the more amplified the response. I'm writing it down. You should be too. You don't just have to fill in the guided notes on your notes, guys. That's not good note taking. Good note taking is listening for other key things I say. Good note taking is writing on those diagrams the way that I do. Okay? Be an active listener. Be an active note taker. Okay? All right, that's enough on that. So, the longer the pathway, the more amplified the response. If this pathway cut off at this step, we would only have 10 to the 2 molecules there. Okay? But the longer it goes on, we get a bigger response down here with 10 to the 8th glucose molecules. Okay? So, the binding of epinephrine causes the glycogen to be broken down into glucose. And we talked about epinephrine a little bit the other day. Remember, that's a hormone. So hormones are going to be, just to review, involved in long distance signaling. Hormones are going to be released by the endocrine system. They travel through the blood. It takes a little bit longer for a hormone to elicit a response, but generally the response will also have a little bit more of a prolonged effect. The response lasts longer than a nervous signal. Okay. And signal amplification, that's what I just mentioned. Each molecule activates more molecules in the following step, and so a small signal can elicit a huge response. One epinephrine molecule binding can cause hundreds of thousands of glycogens to be broken down, okay, or millions even. So one signal molecule can elicit a huge response. That's very important, okay? There's also a lot of coordination of responses. And remember, different cells can have the same receptor but respond in different ways. Okay, that's what we see here. I talked about this a little bit in class as well. The fight or flight response. When you have epinephrine, also known as glucose, that binds, it causes a lot of different responses in the different cell types. So in the eyes, you get tunnel vision. In the heart, you get acceleration. The muscles tense, the stomach slows. It has different responses. All of those cells have the same receptor. Okay, that's our little receptor for the for the epinephrine molecule. But what's different is the relay molecules that they trigger. They have different relay proteins. And so because the relay proteins are different, they cause a different response. Okay? It activates a different pathway with the same signaling molecule and the same receptor. Okay? Now, sometimes you will have a slightly different receptor, but it can't be too different because it still has to fit that epinephrine molecule. Okay? So, let's look at this diagram to just kind of look at some different things that can happen. So you have the signaling molecule that binds to the receptor here that activates the relay molecules and leads to a single response. Sometimes it can activate relay molecules that activates more different relay molecules. And so you have two responses. Okay, we kind of saw a similar thing with the receptor tyrosine kinases. Sometimes you can have another pathway over here that helps regulate that response. So a different signaling molecule is going to activate a different pathway and activate a different protein that could then bind to this protein and activate or inhibit its response. And so that's how two different pathways can affect one another. Okay? And then, and a lot of times medicines will work this way. They will have a different, they will utilize a different pathway to block the activity or enhance the activity of something that's gone a little awry. Okay? And then sometimes you can have, like we just mentioned, a receptor that's slightly different that leads to a different response. Okay? Or we also said you can just have different relay proteins in there that cause a different response. All right. Now, one way to make a cellular response more efficient is through what's called a scaffolding protein. And so you see that this guy here in the pink is our scaffolding protein. And so the scaffolding protein binds 3 different protein kinases. And so when this receptor binds to the scaffolding protein, then it's going to activate all of those protein kinases. So we don't have to go through all of these steps where one signaling molecule has to bind to the other one and to bind to the other one and bind to the other one. And they don't have to kind of diffuse through the cytoplasm to be able to achieve that. They all get activated at once. Okay? And so that makes it a little bit more efficient or quicker of a signal with scaffolding proteins. Okay. And the last thing I want to talk about here today is apoptosis. So this is just kind of an aside from our lesson, but don't misinterpret this to mean that it's not important, because apoptosis is very important. Apoptosis is cell death or programmed cell suicide. And sometimes that's important for cells that ignore signals, okay? We have cells sometimes that just get the signals wrong. They don't respond to them. They get stuck in the on position or stuck in the off position, and they don't, like we talked about with the cholera yesterday. And so generally the body can catch those things. All of these different steps in a signal pathway, not only do they amplify that response, but it causes several safety checks. Okay? At each one of these steps, there's a checker in the cell that makes sure everything is going okay. And the more steps you have, the more opportunities for problems there may be, but also the more checks are in place. And so if a cell is not doing its job correctly, it will get marked for apoptosis. It will get programmed to die. Okay? And so sometimes this happens to bad cells, like we say, but also it's very important in normal cell development. Okay, so when you and I are a wee embryo growing in our mother's womb, we all started out with webbed fingers, okay, or interdigital tissue between our fingers. But these cells get marked for apoptosis, and they start to die. Okay, those cells die, and then you end up with normal non-webbed fingers. Now, some people do have a little bit of webbing. You see it more common in the toes than in the fingers, and those cells just didn't get, fully apoptosis. Other cells, like this one, we see this in cancerous cells sometimes, kind of form these little balls here, and that's called blooding. When those cancer cells are marked for apoptosis and die. Okay, so it's a normal thing. It's sometimes programmed for things that we don't need, like these interdigital tissues, but it's also programmed to prevent bad cells from continuing to divide and replicate and go on and cause problems. Okay, and if those cells ignore those signals for apoptosis, that can cause things like cancer. And we're going to study that more in the latter half of this unit. So this unit is kind of in two parts: cell communication and then cell division, and what happens if these communication signals are ignored. And that's the cancer part. So we'll spend the rest of the week talking about that. You are going to finish up the cell signal transduction pathway worksheet today with Model 2 and 3, and then you can submit that on Google Classroom. And then tomorrow we will jump in with mitosis on Wednesday. And then Thursday and Friday you're going to do some virtual labs at home. So if you have any questions about those, then let me know. You all have a great day.